Hello, thank you for joining us today for an online seminar on cancer research. My name is Liu Chen, and I am a staff field application scientist at 10x Genomics. This online seminar will be about 45 minutes. So today, I would like to first talk about research on elucidating the heterogeneity of the cancer and microenvironment, and then I'll move to the studies on cancer therapy. And I will close by giving you a brief overview of our 10x solutions that are used to support all the research projects that I mentioned today. So cancer is a disease in which cells almost anywhere in the body begin to divide uncontrollably. So a tumor is formed when this unrestricted growth happens in the solid tissues, including organs, muscles, and bones. Tumors may spread to surrounding tissues through the blood and the lymph systems. So the tumor microenvironment is immensely complex and heterogeneous, and the heterogeneity has many dimensions. So for examples, there are many cell types in a tumor. Many of them are therapeutic targets. For examples, immune cells can enable immunotherapy. So in general, a tumor contains these three general cell types. Number one, fibroblasts. Fibroblasts provide supports to the cancer cells. They are immunosuppressive and have been found to recruit regulatory T cells. Regulation factors released from cancer-associated fibroblasts into tumor microenvironment can support tumor growth, angiogenesis, metastasis, and also therapy resistance. The second cell types are the epithelial cells. They line the blood vessels and they supply nutrients and oxygen to the cancer cells. Anti-angiogenic drugs, that is, those that stop the tumor from growing blood vessels, may slow the growth of the cancer, or sometimes it can shrink it. The third general cell types are the immune cells, which are attracted to the tumors. So among the immune cells, T cells, for example, are primary target for immunotherapy. So in this first segment, I'm going to showcase how researchers have used our 10x single-cell gene expressions and the spatial transcriptomic solutions to review tumor heterogeneity. So first is the publications in Cell Reports from Tsinghua University in China. The authors constructed a single-cell network of pre-malignant lesions in early gastric cancers. So they studied the transcriptional landscapes of various cell types across the lesions. The schematic diagram in the middle describes the experimental workflow for the whole study. So mucosa biopsies were collected from various donors. Some used as the normal controls, and some are from early gastric cancers individuals. The transcriptome of single cells were constructed using the 10x single-cell 3' gene expression workflows. For sample preps, biopsy samples were minced to small pieces and digested with the solutions containing PBS and the enzyme collagenase. The cell suspensions was then filtered through 45-micron nylon mesh to remove cell aggregates, and then resuspended in PBS containing FBS. Live cells were obtained by sorting. On your right, you are looking at a TC plot of around 32,000 high-quality cells. Each dot represents a cell. Colors denote annotated clusters identified based on the gene expressions of the known marker genes. From here, the authors were able to identify biomarkers of early malignant cells. Now, other than the single-cell gene expressions, you can also use our Visium spatial gene expression solutions to examine cellular heterogeneity. But here, instead of profiling extracted to purified cells from the tissue, you are looking at cell compositions in its native environment in the tissue. So the Visium spatial gene expression solutions measures total polyadenylated RNA, including mRNA, in intact tissue sections. and then map the data back to the H&E stain image to find out where gene activity is occurring. On this slide on your left, you're looking at the H&E diagnostic images at low magnifications with the tumor regions circled in yellow. Right next to it, on the right, are images with the red represent patches of positive tumor infiltrating lymphocytes. Blue represents the tissue regions with no TIL patch, while black represents the no tissue regions. So tumor infiltrating lymphocytes, or TILs, they are a collection of lymphocytes that have penetrated the stroma of a tumor. They are largely comprises of tumor reactive T cells. Now, from these images, one can imply that donor A may have the potential for a good outcome with the TILs infiltrating the tumor, while donor B does not. You can envision that for TIL therapy, which is a class of immunotherapy, reactive T cells would have to penetrate the tumor to eradicate cancerous tissues, and our Tanix spatial gene expression solutions can provide this clarity. Here is an example from a publication by researchers at the Science for Life laboratory in Sweden. So the authors look at HER2-positive breast tumor microenvironment using the spatial transcriptomics solutions, focusing specifically on dissecting the immune cell infiltrates. They combine cross-sectionings and computational alignment to build 3D images of the transcriptional landscape of the tumor and its microenvironment. Now, shown here on the left is an example of the data where the authors had the H&E stained tissues annotated by a pathologist. So he or she identified these various regions: invasive cancers in red, the normal stroma in green, and then the immune cell infiltrates in yellow. In the middle, you can see the heat map based on gene expressions, showing what the authors call immune scores, which represents the intensity of immune cell-associated gene expressions across the same tissue slice. Red means relatively high expressions, and blue means relatively low expressions. Now, overall, the data shows high intensity of immune expressions in the indicated induced red regions, and that correlates very well with the major immune cell infiltrates as annotated by the pathologists. Now, but as the authors went deeper into this data and broke these signatures down into regions associated with different types of immune cells, they discovered significant heterogeneity. So for example, memory T cells were indeed found primarily in these annotated regions, but other cell types like T helper 2 cells, Th2 cells, they were found more diffusely in the cancerous regions of these tissues, and they are outside the pathologist's annotation. So it is to point out that we were not able to examine this degree of heterogeneity from the H&E stain image alone. Now, the authors also collected multiple sections from one tumor and used those to generate a volumetric reconstructions of the tumor showing the immune cell infiltrates. For the diagram on your right, we are looking at cytotoxic CD8 positive T cell expression signatures in yellow. The 3D data clearly demonstrates the heterogeneous nature of the tumor immune interactions. So overall, this study shows the potential for an improved stratifications and descriptions of the tumor immune interplay, which is likely to be essential in making treatment decisions. Now, as just discussed, our single cell and the spatial solutions offer the perfect system-level approach to study cancer biology. So now, I would like to focus on how our single cell gene expression solutions and the feature barcoding technology can advance research on cancer therapy. In December of 2018, the Science magazine named developmental tracking by single cell sequencing as the breakthrough of the year. So what they are really highlighting is that the many clever approaches and assays combined with the single-cell transcriptome profiling that has enabled researchers to look into biology in a way that they have never been able to do before. Now, as shown on this slide, in a quote from a published paper published in Molecular Cell, the author says that single cell is paving the way for many more discoveries that will come from improved technologies, resolutions, computational methods, and broader clinical settings. So, how can we at 10x help you to perform innovative research to study cancer biology? With our single-cell gene expressions and immune profiling solutions with the feature barcoding technology, you can perform the following. Number one, you can integrate all the information, including the identifications of all cell types that could be influencing tumor growth and progressions, as just discussed. Number two, you can have a deep understanding of immune components and its regulations, which in cell therapy, for example, this includes evaluating the purity and functions of the chimeric antigen receptor T cells or CAR T cells, and studying the mechanisms and monitoring CAR T cells activation upon infusions. Number three, you can also use the solutions to identify drug targets and elucidate antibody components that recognize tumor cells. Each of these application feasibility is supported by many high-profile publications. Now I'm going to highlight some of these publications on cancer therapy and then specifically elaborate on the use of single-cell CRISPRs on immune therapy. Here's an example published in Cell Reports in 2019 by researchers at the McGill University in Canada. So the authors studied responses to a drug targeting the epidermal growth factor receptor, or EGFR, that is used to treat certain cancers, including breast, lungs, and other cancer types. Here, they screened a triple-negative breast cancer patient-derived responder human and mouse xenograft using our 10x single-cell 3' gene expression solution. They generated single-cell suspensions by mechanical and enzymatic dissociations of the tissues using GentleMax. Mouse stroma cells were removed using a mouse cell depletion kit for mutiny. Single-cell suspensions were then washed in FACS buffers containing PBS, FBS, and HIPS, and then stained with PE-anti-EGFR antibody. The cells were then washed, and then cell viability was assessed by staining. Now on this slide, you are looking at a t-SNE plot of about 3,500 cells. Cells are colored by clusters, as marked by the black circle, so the authors identify subpopulations of cells displaying distinct biological features, including a mesenchymal clusters which highly express EGFR that functions as the tumor-initiating cells. Now, these results may have an important implications in therapeutic interventions and to study EGFR-based target treatments in breast and other cancer types. These second examples on cancer therapy research is a publications from the Fred Hutch Cancer Research Center and also additional collaborating institutes in the US. The authors examines the mechanism of resistant response of a tumor caused by Merkel cell carcinoma to immunotherapy treatment. They treated the study subjects with reactive CD8 positive T cells and immune checkpoint inhibitors. And then they monitored the recipients regarding their resistant responses. So for sample prep, the tumor was mechanically minced into small pieces and resuspended in rPMI medium containing DNAs and digestive enzymes. And then the tumor digest was strained through a 70-micron cell strainer. Cells were centrifuged, counted, and assessed for viability, and then resuspended in rPMI freezing medium containing serum and DMSO. Now the cells were stored in liquid nitrogen until use. So when they're ready to do the experiments, the cells were thawed, washed, and loaded into the 10x Chromium platform and 3' gene expressions library was generated and sequenced. Showing on these slides are two identical TC plots constructed with a single-cell gene expression data from the tumor biopsy of one study subject. So you're looking at data of around 7,400 cells. So the cells were obtained during pre-treatment, as shown in the blue clusters on your left, and at the late relapse or the acquired resistance state, shown in orange. The observed separations of the tumor from these two phases indicate substantial transcriptional changes. Now, the plot on your right shows the human leukocyte antigen B, or HLA-B expressions. The red cluster highlighted here is the population of T cells that recognize the oncoprotein, showing that they are prevalent during the response phase. So why the observed resistance then? So what this author found was the tumor had transcriptionally shut off the HLA expressions, and that although the T cells could recognize the antigen, it could no longer be presented. So note that the results imply HLA-A downregulations, not HLA-B. Now, and this cannot be distinguished by immunohistochemistry. Now, what is particularly interesting with this finding is that transcriptional HLA repressions is reversible, and it could be the basis of prolonging response to immunotherapy in patients who become resistant by this pathway. Now, resistance to immunotherapy is not uncommon, and resistance is what was found to happen in the study subjects about 1 year after the treatment began. But although we know that resistance is a commonplace, we are only beginning to understand the mechanisms behind the resistance. Many other publications have reported the loss of the CD8 positive T cells that are supposed to initiate the immune response. But in this case, in this study subject, the T cells persisted. So this wasn't the answer, and no DNA mutations were found to explain the resistance. The answer lies in the suppressive of the specific HLA gene that present the targeted viral epitope. In this second segment, I would like to talk in more details about CRISPR and immunotherapy. Now, CRISPR-Cas9 is a gene editing technique that shows potential in cancer immunotherapy. So for the last 2 decades, targeted therapies that use drugs to target cancer cells have been the standard treatments for cancers. So for example, earlier on, I talked about examples from publications studying response of a drug targeting EGFR that is used to treat breast cancer. Now, one of the most newest and the potentially most promising areas of cancer research and treatment is immunotherapy, which harness the patient's immune system to attack tumors. So cancer cell therapy begins by collecting immune cells from the blood. These cells are then modified. For example, in CAR T cell therapy, T cells are genetically engineered to express a CAR specific for tumor antigen, followed by cell expansion, and then they are returned to the affected individuals to spur his or her immune system to more effectively target the disease. So T cell modification can be done through viral-based or non-viral-based gene transfer methods, or by CRISPR-Cas9. So what is CRISPR? CRISPR stands for clustered regularly interspersed short palindromic repeats. Now, Cas9, on the other hand, evolved as a bacterial immunity, but now are leveraged to allow for a sensitive and sequence-specific targeting of genetic perturbations through programmable guide RNAs. So guides are transfected or transduced into cells, then complex with the Cas9 protein to seek out and to disrupt the targeted genes. So CRISPR-Cas9 is adaptable to many cell types, including cancer cells, organoids, and primarily immune cells. This technique is incredibly specific, but the design is flexible and it has been widely used across various applications. So CRISPR screens allows you to profile thousands of gene targets simultaneously with the goals such as to identify novel drug targets and to accelerate hit identifications and lead optimizations. Number two, optimize the current chemotherapies with enhanced functional genetic screens. Number three, to identify genes that make cells resistant to specific drugs. Number four, to screen genes that are essential to cancer survival. And number five, and the last, it allows you to identify molecular and cellular mechanisms of disease pathways and therapeutic targets in cell lines models or in organoids. Now, however, in traditional CRISPR applications, the transduced cells still require a long culture time, and traditional assays only allows for crude phenotypic readouts based upon the survival or the proliferation assays once a selection is applied. So one of the key benefits of the single-cell CRISPR screen using the 10x technology is a much faster turnaround time from the initial start of the experiments to the time when you can look at your data. So the way how our method works is you take your cell populations of interest, so whether an in vitro screens or from an in vivo model, and transduce your CRISPR system of choice into the cells. After allowing a few days for the CRISPR-driven perturbation to take place, for examples, by culturing the transduced cells for 2 to 3 days, you can then harvest your cells using a 10x-compatible protocol that works with a sample type, and then you can prepare them for single-cell transcriptomics analysis. The 10x feature barcoding technology is engineered to read barcoded guide RNA, giving an easy way to identify which cell received what perturbations. So our approach has a number of advantages. So it is fast because the readout is transcriptional phenotype. Your assay is only as long as it takes for the gene expression changes induced by your perturbations to work their way through the entire cell population. Number two, it is higher in resolutions. The readout is full transcriptomes rather than a limited phenotype like growth or a specific reported assays. Number three, you have a reduced batch effects. For example, treatments that are cells that receive the guide RNA and the control cells that did not, they can be pooled in the same experiments and they will get prepped together. Number four, the assays is programmable. So this comes with CRISPRs. You can target any genes that is accessible to CRISPR, and you can tell which cells got what treatment versus those that did not with the transcriptome readout, rather than having to do a deep sequencing. Number five, the assay is scalable due to the fast runtime of the Chromium systems. Number six and last, target and samples can be multiplexed. That means you can screen many targets in one run and map the guide to individual cell easily. In these publications in 2018 in Cell by researchers from the University of California and the US, the authors used CRISPR in the context of drug target discovery efforts, specifically on cancer immunotherapy. So the Cas9 ribonuclear proteins can be delivered to primary human T cells to effectively knock out checkpoint genes, or even to rewrite endogenous genome sequences. Now, while the deletions of the canonical checkpoint genes encoding PD-1 may enhance T cell responses to some of the cancers, an expanded set of targets would offer additional therapeutic opportunities. For example, to enhance T cell proliferations and productive effective responses upon stimulations. Now, in addition, in the real tumor environment, immunosuppressive cells and soluble molecules can accumulate within the tumors and therefore hamper productive anti-tumor T cells responses. So gene targets that influence a T cell's ability to overcome such immunosuppressive tumor microenvironments could extend the reach of the adoptive cell therapies to solid tumors. So here, the authors validated a large-scale CRISPR screen and then identified perturbations that enhance cancer cell killing and identify regulators of stimulation responses in the primary human T cells. So in this study, human T cells from 2 donors were transduced with 48 single guide RNAs. The transduced cells were electroporated with the Cas9 proteins and subjected to single-cell transcriptome analysis either with or without re-stimulation. So the goal is to characterize alterations to cell state and stimulation response. Now you can cluster your populations based on their overall perturbed transcriptomes and then correlate the gene expressions with the guide RNA expressions. So the UMAP plots on the top give you an idea of the high resolutions of these screens. So the first plot on the left at the top show all sequence single cells with identified single guide RNAs across resting and the re-stimulated T cells. The middle plot at the top shows the scaled gene expressions for 4 genes indicating cluster associations with the processes that are critical during the adaptive immune response. The rightmost plot on the top shows the unsupervised clustering results of single cells based on gene expressions, and in total, 13 clusters were identified. The heat map at the bottom shows the average gene expressions across stimulated cells with different single guide targets. It shows how 7 guide RNAs can perturb regulators involved in cell cycle, T cell activations, and other processes critical for immune response. So the authors confirmed that knockout genes led to the perturbations of genes involved in proliferations and cell death, and a subset of the hits enhance in vitro anti-cancer activity in human T cells, suggesting that this screening platform could be used to find promising preclinical candidates for next-generation cell therapies. Now, I would like to emphasize 3 points that are critical to these projects. Number 1, the loss of functions CRISPR screening with the single-cell resolution readouts creates completely and ambiguous relationships between the knockout and the biological impact. So in this case, the authors chose to find targets with impacts in CAR T-cell immunotherapy. Now, many researchers have promised to speed up the drug discovery process, but processes like this, which combine 2 of the most powerful tools in biology, CRISPR and single-cell sequencing, are actually delivering on that promise. So number 2, the authors shows a pipeline that is relatively easy to execute and substantially more informative and scalable than previous iterations of the technology. Here, they shows that the pipelines can identify targets, and once they are knocked out, they positively impacted primary T cell proliferations upon T cell receptor activations and induce cancer killing capability. Number 3 and last, most of the critical biology cannot be reproduced in immortalized cells, and that screening methods based on primary cells are ideal for the 10x technology. Now, also to note that the full workflow only took 9 days after transductions. So the authors had saved time with the reductions in the survival culture period because the readout and assessment of the perturbations were directly informed by the rich data around transcriptional landscapes associated with each of the knockout. Now, the traditional way of doing CRISPR screen is that cells are transduced and then selected with an antibiotic or fluorescent marker after 2 days or so. And then the cell populations get some kind of treatment to either drugs, small molecules, or other stimulants, and they have to be kept in culture for about 2 weeks to see an effect. Then the cells get selected, and those that survive the treatment will do so because the guide RNA specific to that cell confer resistance. And then another round of selection is to be conducted to select all cells with guide, each with a single guide RNA, and you have to have a culture running in parallel as the negative control. Finally, you can then assess your crude phenotype before you isolate the guide RNA, perform PCR amplifications, and then use deep sequencing to assess the guide RNA content. Now, using the 10x strategy compared to the traditional methods, there are 2 key steps in the workflow that are substantially reduced in time. Number 1, after stimulating your cells, you will only have to keep them in culture for 2 to 3 days before running them through the 10x assay, because it only takes a few days to see the impact of a guide RNA to the perturbed transcriptomes. Another advantage is that the workflow is applicable to a wide range of primary cells which usually cannot be kept in culture for more than a few days. And there is no need to select specific cells from the populations and assess the phenotype, because single-cell resolution will allow you to distinguish all individual cells, and there is no need to do deep sequencing to identify the guide RNA. It only takes as little as 5,000 raw reads per cell to identify the guide. Now, in these publications in Cell in 2019 by researchers at Yale University in the US, the authors used single-cell CRISPR analysis to narrow in on immunotherapy targets. So they started with a genome-scale CRISPR screening library and performed both in vivo and in vitro assays. So the in vivo assays involve transferring T cells from a Cas9 mouse into another tumor-bearing mouse and test tumor infiltrations. The in vitro assays involve T cells from the same Cas9 mouse tested in a degranulation assay. Now, combining these two screens led the team to a short list of hits. In particular, DEAH box helicase 37 or DHX37 appear as a hit. So this target has not been previously associated with T cell functions. So the authors then used our 10x single-cell gene expression assay to investigate the transcriptome of DHX37 CRISPR-modified tumor-infiltrating lymphocytes. They found that the most highly upregulated genes in the DHX37 knockout cells were indeed associated with T cell therapy, T cell potency. So they then follow up on literature searches to study DHX37 activity in human T cells and identify it as a novel high potential target. Now, shown here is a great science paper that was published in February 2020 using our assay as a QC methods to evaluate the purity and functions of T cell receptor engineered T cell or TCR-T. So these cells were made by the authors themselves before infusing them into the study subjects. So that is, the authors took T cells from the study subject, make a series of edits to improve the ability of the T cell to fight cancer, and then infuse them back into the subject. They then monitor the fractions and phenotype of the infused T cells and their evolution over time in the study subject. Now, in this landmark study published by researchers at the University of Pennsylvania and Stanford in the US, so the authors make a total of 4 edits to the T cells. So they use CRISPR to disrupt the endogenous T cell receptor alpha and beta genes to reduce TCR mispairing or T cell receptor mispairing, and to enhance the expressions of a synthetic cancer-specific T cell receptor transgene. So the authors also use CRISPR to disrupt the PD-1 receptor to suppress the immune checkpoint interactions. And then they introduce an engineered T cell receptor that recognizes unknown cancer-specific antigens. So there are many key questions about the infused T cells in the patients. Do the engineered T cells persist in the patients, and for how long? Do they have the intended edits, and at what rate? And what phenotype do the transferred T cells adopt? How does the immune system of the study subject respond to the engineered T cells after infusions? So the 10x Genomics single-cell immune profiling solutions enables to sequential answers to all of these questions. That is, we have a platforms that unify diverse questions under a single experimental method. So the results demonstrate that CRISPR-Cas9 gene editing technique is a powerful tool for cancer cell therapy. And here is the first in-human phase 1 clinical trial to test the safety and the feasibility of the CRISPR-Cas9 editing to engineer T cells. Now on this slide, you're looking at the results from the single-cell 5' gene expression sequencing of a study subject with the CRISPR-Cas9 engineered T cells during pre-infusion, that is day 0, and post-infusions on days 10 and 113. So the plot on your left shows the proportions of the pre-infusions and post-infusion T cells without mutations. That is the wild type, those with the T cell receptor alpha and beta genes and the PD-1 mutations, and those that express the T cell receptor transgenes. Here, the authors shows that they are able to measure the fractions of cells carrying each mutations in the infusion products and also in circulating blood over the entire treatment process. Now, the fractions of edited cells declines somewhat but appears to reach a steady state. Now, the persistence of the transduced cells marked by the blue line is remarkably stable for months after infusions, suggesting that immunogenicity, that is, to provoke a native immune response against the knock-in T cells, is minimum under these conditions. And this demonstrates the feasibility of CRISPR gene editing technique to be used for cancer immunotherapy. Now, the UMAP plots on your right shows the expression data. The analysis was performed on all T cells across time points, but shows only cancer-specific T cell receptor transgene expressing T cells. So the increase in TCF7 expressions is indicative of an acquired central memory phenotype rather than an exhausted phenotype, which may have enabled the engineered T cells to remain for a long term. Now, it is conceivable that the industry can scale up and do even more this kind of study. So more therapeutic and genetic engineering targets, more clinical trials, and more lives hopefully can be saved. Now, in summary thus far, I talk about the study of tumor heterogeneity using our single-cell gene expressions and the Visium spatial transcriptomics solutions. I talk about using single-cell gene expression to study cancer drug response and to elucidate the mechanisms of resistance to immunotherapy treatment. And using CRISPR as part of our 10x feature barcoding technology for gene expression study on the context of drug discovery, narrow in on immunotherapy targets, and then showcase a phase 1 clinical trial to test the safety and the feasibility of engineered T cells for immunotherapy. Now, in this last segment, I'd like to talk about the 10x solutions behind all these great results that I presented today. With the Chromium single-cell platform, you can profile tens of thousands of cells or nuclei and multiple samples at one time to reveal the full cellular heterogeneity on the transcriptomes and the epigenome levels. Shown in this slide is a simplified view of our single-cell gene expression workflow. So starting with single cells or nuclei, sequencing libraries are generated using the Chromium controller and the single-cell reagents. The single-cell reagents and consumables include the gel beads. The gel beads are our primer delivery systems, the microfluidic chips for partitioning cells or nuclei, and the library construction reagents. The workflow generates libraries that are ready for short-read sequencers. After sequencing, the results are analyzed using our Cell Ranger pipeline and visualized using the Loop browser, both of which are available from download from our 10x website. Showing on this slide is a schematic representations of the Chromium single-cell 3' version 3 gene expression gel bead. Now, each gel bead contains many copies of the 3 types of oligos with the unique capture sequence. So the first contains a poly-DT stretch that enables the capture of polyadenylated RNA molecules, including mRNAs. So you can use these beads to capture mRNA as much higher sensitivity. So the oligos also contain the 10x barcodes, which identify the particular cell or the nucleus in which the transcripts or the feature is found. In addition, there is the unique molecular identifier or the UMI, which is used to identify the particular molecule that is present in your library. So essentially, the UMI is to tag each unique cDNA that is synthesized from each polyadenylated RNA molecule. To enable the feature barcoding technology, 2 additional oligos are present on the gel beads, which contain capture sequences for the features of interest. Now, these second and third oligotypes are to capture any feature that has a DNA tag with a sequence that is complementary to the capture sequence. On the gel beads, 2 different capture sequences are present, either capture sequence 1 or capture sequence 2. Now, 2 capture sequences provide more flexibility in experimental design. So what are these features? So these features can be oligonucleotide conjugated antibodies, if you're looking at cell surface protein profiling, or they can be small guide RNAs for CRISPR experiments. So all in all, the Chromium single-cell gene expression solutions with the feature barcoding technologies combines the feature detections with the unbiased readout of the transcriptome from each single cell. Our immunoprofiling solutions allows you to assess the transcriptomes and the full-length VDJ gene sequences from the pair alpha and beta chains on the T cell receptor sequences from individual T cells, and also the pair heavy and light chain immunoglobulin or B cell receptor sequences from individual B cells, and both allow for full isotype resolution. The feature barcoding technology is also enabled on the immune profiling solution. That means you can measure 5' gene expressions, T cell receptor sequences, B cell receptor sequences, cell surface protein expressions, and then you can link the full-length pair T cell and B cell receptor sequences with the antigen specificity. Now, our VCM spatial gene expression solutions is the first commercially available solutions for generating spatially resolved gene expression profiles. So these applications allows you to see not just histological images of tissue sections, but also allows you to analyze a deep molecular profile describing the gene expressions across the entire tissue section. So on the gene expression workflow slide, there are 4 capture areas with approximately 5,000 spots in each. There are millions of capture probes per spot. Now, the oligos on the probes contain a poly-DT stretch that enables the captures the polyadenylated RNA molecules, and the oligos also contains the spatial barcodes, which identifies the locations of where the transcript is found on the tissues. The oligos also contains UMI, which is used to identify the particular molecule that is present in your library. So similar to how it is being used in a single-cell gene expression assay, the UMI is to tag each unique cDNA that is synthesized from each polyadenylated RNA molecule. So depending on the tissue type and the native cell size, the current design configuration allows you to obtain the unbiased transcriptomes from 1 to 10 cells per spot. Showing on this slide is the VCM spatial transcriptomics workflow. It starts with cryosectioning the frozen and OCT embedded tissues and placing them onto the capture areas on the slide, followed by tissue fixation, H&E stain, and then capturing the H&E stain image. After that, the tissue is permeabilized and the RNA diffuses out from the tissues and binds to the capture probes on the surface on the slide, and then cDNA synthesis is performed. The library is sequenced using a short-read sequencer. During data analysis, spatially barcoded cDNAs are mapped to the H&E stain image using the unique spatial barcode sequences. So our spatial gene expression profiles link solution, our spatial gene expression profiling solutions combines the benefits of traditional histological technologies with the massive throughput of RNA sequencing. So the ability to layer on gene expressions informations provides a clearer pictures of the biology captures on the tissue slide. With that, thank you for listening to this online seminar. Have a great day.